Welcome back to this next video, and this is the part 2 of the sex-linked inheritance. In the first part of this video, I've told you about the X-linked dominant traits, and in this video we are going to talk about the X-linked recessive traits. Now the X-linked recessive traits, they are those traits in which two copies of the mutant allele they are present. You have both the copies of the mutant allele on your chromosomes. For example, if you talk about the hereditary pattern in which a recessive gene on X chromosome result in the manifestation of trait in the male offspring and a carrier trait in the female offspring. For example, you have one mutant copy of the allele. So if you talk about the males, and if the male they are getting that mutant copy of the those particular allele, the males they are going to be affected, because males have got only a single X chromosome, and if that X chromosome is carrying a mutant allele, that means that the males they will be affected. If you talk about the females, if they have got two X chromosome, if on one X chromosome they have got the mutant allele and on the other X chromosome they have got the normal copy, we usually call them as the carriers, and they do not manifest those particular traits. Those particular traits are not presented by the carrier females. Now that means that if you talk about the X-linked recessive diseases or trait in females, so in females, this particular phenotype will be expressed when two copies of the mutant allele are present. So if you talk about the males, male have got a single X chromosome, and if they are getting one copy of the mutant allele on their X chromosome, the only X chromosome in the male, they are going to show you the disease. But if you talk about the females, if they have got one mutant copy and one normal copy, they will be the carrier, and the female will be affected only in this particular case when the both of the X chromosome they carry the mutant allele. This is the case where the females they will be affected, and in this particular case when they have got one normal copy on one of the X chromosome, they will be the carrier and they will not be showing that particular phenotype. If you talk about the characteristics of the X-linked recessive traits, all male offspring or all males possessing an X-linked recessive mutation will be affected. Male have got a single X chromosome, and if that X chromosome is carrying a mutant allele, all those males are going to show the X-linked recessive mutation and they will be affected. If you talk about the females, all offspring of a carrier female, if you have got a carrier female, all of the offspring they will be having a 50% chance of inheriting the mutation. For example, you have got carrier female over here. On one of the X chromosome they have got the normal allele. On one of the and on the other X chromosome they have got a mutant allele. If you cross them, this X is for crossing. If you cross that with an normal male, this is not small a, this is capital A. If you cross that with a normal male, when you cross them, you can actually see is that this particular son, it is going to be affected, and this particular female, she will be the carrier, because she is having one normal copy and she is having one mutant copy. So all offspring of a carrier female, they will be having a 50% chance of inheriting the mutation. So you have got 4 offspring, 2 of them they are inheriting the mutations, 2 of them they are not inheriting the mutation. Therefore you can say that all offspring of a carrier female, they will be having a 50% chance of inheriting the mutation. If you talk about the all female children of an affected father will be carriers. If you talk about the females of an affected father, all of the female that will be the carriers. For example, you have got a normal mother, they have got both normal copies of both of the alleles, they are normal, and you have got an affected father. The father have got a single X chromosome, and the allele present on it, that is the mutant allele. So when you talk about the female children of this affected father, all of them, all of the female offspring, they will be the carrier. This is one female offspring. they are getting one normal copy of the allele from their mother and one of the affected allele from their father. So these males, they will be the carrier. This female, this will be the carrier. And if you talk about this female again, this female will be carrier again because she is getting one normal copy from the mother, one affected copy from the father. So all the females, children of an affected father, they will be the carrier. If you talk about the males, no male child of an affected father will be affected. The father is affected, the mother is normal, no male child will be affected. The reason is the males, they are getting the X chromosome from their mother and not from their father. And if the mother is carrying the X chromosome and mother is carrying a normal copy of the allele on their X chromosome, all of the sons, they will be getting a normal copy of the alleles and they will not be affected. So if you see in this cross, all of the males, they will be normal and all of the females, they will be carriers. So the female children of an affected father, they will be the carrier, and the male children of an affected father, they will be normal. We will be taking the hemophilia as an example for the X-linked recessive traits. Now what this hemophilia means? This hemophilia is made up of two words. This hema means blood and this philia means love. This means love for the blood. This hemophilia is a group of the hereditary genetic disorders that impairs the body's ability to control blood clotting or coagulation. When there is cut on the blood vessel, the normal body is going to make blood clots, and that blood clot is actually going to stop the bleeding when there is cut on the blood vessel. But in the hemophilia, this clotting or this coagulation, it is impaired. That means that the blood vessel, that the bleeding, that will continue for a longer period of the time. So in hemophilia, usually in hemophilic patients, the body's blood clotting or coagulation system, that is impaired. There are two major types of the hemophilia when you talk about the X-linked recessive disorders. One of the hemophilia that is called is the Hemophilia A, and it is the most common hemophilia because 80% of the cases throughout the world, they are of the Hemophilia A. In this Hemophilia A, there is a clotting factor which is called is the clotting factor 8. We will see that in a while. So in the Hemophilia A, this factor 8, the clotting factor 8, there is a deficiency of this particular factor 8. And if you talk about the hemophilia A, I've told you this is the most common form. So it is present in about 1 in 5,000 to 10,000 male births. So out of 5,000 to 10,000 male births, one of them is hemophilia A patient. If you talk about the hemophilia B, a less common type of the hemophilia, because only 20% of the cases of hemophilia are hemophilia B around the globe. In this hemophilia B, there is a deficiency of factor 9. And if you talk about the numbers, there is hemophilic B patient, one hemophilic B patient in about 20,000 to 34,000 male births. Let us discuss about the blood clotting mechanism in a little bit detail, how this blood clotting mechanism that is actually working in a normal body. Now the gene for the factor 8, say for example we are talking about the hemophilia A, so the gene for the factor 8 that is present on the X chromosome, therefore we call them is the X-linked disorders. Now in the normal condition, when there is no cut on the blood vessel, for example, this factor 8 is in association with another glycoprotein in the blood which is called is the von Willebrand factor. So in normal condition, this factor 8 is in complex with the von Willebrand factor. And in this particular form, when the factor 8 is in complex with this particular protein, the factor 8 is actually in the inactive form. That is not going to initiate the process of the coagulation. But when the signal arrives, for example, there is a cut on the blood vessel, that signal actually is going for the activation of the thrombin protein, which is also called as the factor 2A. So when in response to the signal, the thrombin or the factor 2A is activate, what this thrombin is doing, that it is going to dissociate this factor 8 from this particular complex. So this thrombin is actually breaking this particular association of the factor 8 and the von Willebrand factor. So when the factor 8 that is dissociated from the complex, this factor 8 is actually a cofactor for another coagulation factor or coagulation protein, which is known as the factor 9A. So when the factor 8 it combines with the factor 9A, the factor 9A it gets activated, and when the factor 9A gets activated, it is going to activate another factor which is called as the factor 10. When this factor 10 it is activated, it combines with its cofactor which is called as the factor 5A. And when the factor 10 it combines with the factor 5A, you are actually getting this particular complex of the factor 10 and the factor 5A. This complex is going to activate more thrombin in the body. So initially you have got a very low number of the thrombin. Now you are activating a lot of thrombin in the body, so as you can go for the coagulation of the blood. What this thrombin is doing, that it is going to cleave the fibrinogen, and when the fibrinogen is cleaved by the thrombin, it is converted into the fibrin. Now when the fibrinogen it is converted into the fibrin, many fibrin monomers they combine with each other to make polymers. I'm showing those polymers over here in the form of the lines. So these are the fibrin polymers that have been made by the cleavage of the fibrinogen by the thrombin protein. So when the fibrin they combine with each other, they are making these polymers. If you look at this particular structure, it is, you can say, a very weak kind of the structure, and it will not be able to stop the bleeding. What the body do is that this fibrin polymers, they are cross-linked by another factor which is called as the factor 13. Like these lines, these horizontal lines, they are showing you the fibrin polymers. I'm showing that over here. And these vertical lines, they are actually showing you the factor 13, which is cross-linking these fibrin polymers. Now, if you compare these two structures, this is a very stronger kind of the structure, and it can effectively stop the bleeding. And this fibrin polymer, when it combines with its cross-linker, the factor 13, this is actually the blood clot that stop the clotting, that stop the bleeding in the body. Now, if you look at the factor A, the hemophilia A, for example, so if you have got a deficiency in the factor 8, that means that all of the process below this line, that is going to be affected. So in the end, you will be actually having problem in the formation of this particular blood clot. So in fact, in hemophilia A, there is a deficiency of this factor 8, thereby affecting the whole process of the blood clotting. When you talk about the hemophilia B, there is a deficiency of this particular factor 9, and when there is a deficiency in the factor 9, that means that all of the process below it that is going to be compromised, and in the end there will be a problem in the formation of the blood clot. Like most recessive X-linked disorders, hemophilia is more likely to occur in males than females. Because the male, they have got one X chromosome, and if that X chromosome is getting a mutant copy, that means that the male will be hemophilic. When you talk about the females, the female have got two X chromosome, and they should get two mutant copies on both of the X chromosome in order to show the hemophilic phenotype, which is actually a difficult task to get two mutant copies on both of the X chromosome as compared to mutant copy on one X chromosomes. Now, although it is not impossible for a female to have hemophilia, but it is very unusual. Now, this is not the case that you will not find any hemophilic female, but this particular phenomena, it is very unusual. And one of the scenario in which female can be hemophilic will be this one, that if the father is hemophilic and the female is a carrier, and there are 50% chances that the female that will be hemophilic. For example, if you talk about this hemophilic male, the genotype will be this one. If you talk about the carrier female, the genotype will be this one normal copy and one mutant copy. And when you make a Punnett square, you are actually having this one, this particular female, she will be hemophilic because she is getting one X chromosome from her mother, which is carrying a mutant allele. The father is having only a single X chromosome, which is having a mutant allele. And when these two chromosomes, they combine with each other, in this particular case, the female, she will be hemophilic, although the other female, she will be the carrier because she is getting one normal copy from the mother. If you talk about the bleeding time or the bleeding intensity, so a hemophilic that does not bleed more intensely than a person without it. Now there is no change, there are no differences in the intensity of the bleeding in hemophilic and non-hemophilic persons. But the hemophilic person, they bleed for a much longer time, and if they are bleeding for a much longer time, that means they are going to lose more blood as compared to the non-hemophilic one. Now in severe hemophilics, even a minor injury can result in the blood loss lasting for days or weeks, or even never healing completely. So if you are not able to make an effective blood clot, that means that you can lose blood for days or for weeks, or if that is a very severe kind of the damage, that can even never heal completely. Now, if in areas such as the brain or inside joints, this can be fatal. If you are losing blood for days or for weeks in your brain or inside the joints, this can be fatal or permanently debilitating.